speaker?
Yes, just adjusted. So this is more convenient time.
Right. Yeah.
Including many circumstances here, taking into account many circumstances here.
Right.
Hello and good evening. It is a very fortunate case that we continue the inspiring online seminar series of TÜBİTAK Research Institute for Fundamental Sciences, which we organize for national and international audiences to mitigate the negative impacts of COVID-19 situation in the world on scientific thought and its creation. Now, it is my pleasure to welcome all of you for this occasion of our interdisciplinary seminar series. All of you joining us through the Zoom platform and all of you watching us through all social media platforms of TÜBİTAK, the Scientific and Technological Research Council of Turkey. It is also my distinct pleasure to introduce you our tonight's very special speaker, Professor Achillefs Kapanidis from the University of Oxford. He's going to give a great talk entitled Unlocking Gene Expression Mechanisms and Detecting Viruses via Single Molecule Imaging. At the end of the talk, we will have a question-answer session where questions can be asked by sending a message through the chat button of the Zoom platform or just by raising hand. Achillefs Kapanidis is a professor at the Department of Physics of Oxford University. After completing a degree in chemistry at the Aristotelian University of Thessaloniki, Greece, Achillefs Kapanidis obtained a master's degree in food chemistry and his PhD degree in biological chemistry, both at the Rutgers University, New Jersey, USA. After holding research scientist positions in a single molecule biophysics at Berkeley, Lawrence Berkeley National Lab, and University of California, Los Angeles, he started a research group as a senior lecturer at Oxford University in 2005 and became a professor of biological physics in 2013. In 2011, Professor Kapanidis became a fellow of the European Research Council, and in 2016, he became a Wellcome Trust investigator. Professor Kapanidis is currently leading a group of physical and biological scientists known as the Gene Machines Group. The group studies biological machinery involved in gene expression, maintains, regulation, focusing mainly on gene transcription and DNA repair. The main tool of the group is single molecule fluorescence microscopy, a technique that measures nanometer distances and molecular interactions in real time, in vitro and in living cells. The work of group is multidisciplinary, combine optics, photonics, imaging, biochemistry, molecular biology, modeling, and signal processing. With this, I want to thank once again Professor Achillefs Kapanidis for joining us for this seminar. We are really looking forward to it. Achillefs, you're welcome to your talk, please.
Thank you for your kind introduction, Alkram. It's a pleasure to be part of your series. Thanks for reaching out, and in these very difficult times for the entire planet, and of course for our countries, to actually have the opportunity to discuss our work with colleagues in Turkey and start a positive relation that I can also continue with time. So let me share my slides to start the talk. I'll share the screen and hopefully, share screen. Okay, hopefully you'll be able to see my first page. Okay, that's great. Okay, so I hope you can see
It's perfect.
the first page. Okay, fantastic,
perfect.
fantastic. All right. So today I'll be telling you a few of our latest stories on the use of single molecule detection, the ability to.
Achilles, I am sorry, but bottom
Yes.
of the page is not seen indeed.
Oh, okay. That's. Let's see whether we can fix this. So you cannot see the date, or
yeah, bottom there's a line. I see. I guess that there's a line.
do you see online Do you see online seminar?
Yeah,
TÜBİTAK
not seen indeed.
Ah, okay. All right, that's not good. Let's try this again.
Oh, okay. Now.
Okay, you see it now. Oh, I think maybe the screen.
You should share a screen again.
Yeah, let's do that. Okay. Does it work better?
Now it is perfect, really.
Okay, fantastic. All right. Okay, after this false start, let's continue. And yes, today I'll be telling you how we use the power of being able to image individual molecules, fluorescent molecules, to look at biological mechanisms that are centered around gene expression, the process that takes us from the level of DNA to the level of proteins. And I will be discussing about the mechanisms that allow the RNA polymers, the machine that copies DNA into RNA, to open the DNA in order to read the information that is stored in genes, and also how we can use the ability to detect single molecules to be able to follow single molecules as they perform their function inside living bacterial cells, and then how these methods can be repurposed to actually detect viruses, including coronavirus, very rapidly and specifically. So we have an interdisciplinary group at Oxford that makes this work possible. Again, physical and biological scientists work together to study the machinery of gene expression and DNA repair. We're focusing in particular on the workings of nucleic acid polymerases, and as I mentioned to you, the main technique that we're using is single molecule imaging, and we complement these measurements with conventional biochemistry approaches. So when we perform measurements in vitro, we look at conformational changes that are taking place either on the DNA that's being processed or the protein that is performing the function. And we'll see examples of this shortly. We also perform experiments in living cells where we can monitor the motion of molecules inside bacteria, or we can use the power of localizing individual molecules with very high precision to generate high resolution images of biological structures in cells. In order to perform these studies, we built our own single molecule microscopes, and we have also worked on miniaturizing our instrumentation. And in fact, we have even spun out a company that makes a miniaturized single molecule fluorescence microscope called the Nanoimager. Now, equipped with the understanding of the biomolecules, the molecular mechanisms, and the instrumentation and software, we're also developing biosensors and assays of diagnostic value. So I will be focusing today's talk on the mechanisms of transcription, at least the first two parts. So what is transcription? Well, this is the process that allows RNA polymers to read the information of DNA converted into RNA. So that happens during a transcription event. A gene becomes available for the binding of the polymers, starts making RNA. If the RNA is translated by the ribosomes, it can form a protein, or it may be a stable RNA, ribosomal RNA, so it will become part of the ribosome. Of course, this is a cartoon version of the process. If we look at the specific case of bacterial transcription, we'll see a complex pathway of binding events and conformational changes. So the machine that will perform transcription will first bind to a protein that will lead it to the right place for the copying of the DNA. And after this initial binding event, the DNA will go through conformational changes. We'll discuss this in more detail in the next few minutes. And there is a transcription bubble, an open part of the DNA that is formed during the process. And when the nucleotides for the synthesis of RNA are present, then the machine will start making short RNAs. This often tend to dissociate, and the system resets back to the open complex. And it does this a few times till it manages to make an RNA that is longer than a certain length, and then moves from this initial mode of transcription to the elongation mode of transcription. So this is an example of a pathway that is mirrored in other genetic processes, in fact, in other processes that involve protein interactions with substrate molecules. So in our system, the highlight structures that you see here have been resolved with high-resolution structural biology methods, X-ray crystallography, or electron microscopy, but a lot of steps that have multiple transitions, or they have multiple complexes that is very difficult to synchronize or to isolate, and they have not been resolved with structural biology methods. So almost 20 years ago, we started looking at this mechanism with the ambition to actually see it from the perspective of an individual RNA polymerase, individual protein, or individual DNA that's being processed. And this way, we'll have unique access to the process and the paths and the transitions and the kinetics of all these changes as they happen. And therefore, we can solve the question is how you put all these structural snapshots together, these are snapshots that you get from the structural biology methods, in order to understand how a process actually works. So you can see here that we have all these pictures of a galloping horse. So what is the sequence of events? Of course, you can cheat and then look at the numbers here and then put it in the right sequence. But the idea is that to use our unique single molecule tools that give you access to this trajectory to put these photos in the right sequence and then to play them as a movie so you can visualize the process. And the way we do this at a single molecule level is to identify sites in the machine or the substrate that participate in conformational changes, and then try to visualize these conformational changes and how fast they occur, and what is the sequence of the motions that are taking place. And the tool that we use to do this is an interaction between fluorophores that is known as fluorescence resonance or first resonance energy transfer that works essentially as a ruler, dynamic ruler for the nanoscale. So in this ruler, you need two fluorophores, one that acts as a donor and another as an acceptor. And when you excite a donor, if the acceptor is in close proximity, within a few nanometers, some of this excitation energy is transferred to the acceptor. So when you visualize the emission from these fluorophores, you see low fluorescence from the donor and high from the acceptor. Now, if there's a conformational change that moves the acceptor further away, the efficiency of the FRET drops, and now you see mainly the fluorescence from the donor. This is a process that's quite well understood in terms of distance dependence, and therefore by measuring the energy transfer efficiency, we get information about the distance or between the two probes, and the dependence of the efficiency to the distance has this very steep dependence because of the power to the sixth here. And you can see that there's a characteristic distance that's associated with 50% FRET efficiency, and then you're very close, you have very high efficiency. When you're far away, you have zero efficiency. And then you have this nice range that you can use to look at distances. So now if we zoom in at a certain part of this transcription process, like to basically focus on the part of the pathway where the machine opens the DNA to start reading it. And we'd like to understand what is the sequence of motions here. Now, this is an important process. It's important for understanding how transcription is regulated. If you want to model transcription, you have to understand the kinetic steps that are taking place. And also, it's a point where antibiotics can stop the RNA polymers of microbes and therefore act as antibiotics that kill bacteria. And of course, understanding this process may be useful to do this in higher organisms. So here's a puzzle that we're trying to solve here. RNA polymers has this cleft, this opening that is fairly narrow. So there are these two jaws that delineate a channel in which the DNA needs to bind. But if you look at the width of the DNA, it's actually wider than the opening of this channel. So in order to fit the DNA in this channel, something has to give. The channel needs to open, or the DNA needs to be deformed in some fashion in order for the loading to take place, or both of these events. And that's the puzzle that a brilliant postdoc in my lab, Abhishek Mazumdar, is trying to solve. Now, understanding the mechanism will allow us to see which of the two models that describe the process are correct. There's a model that basically says that the mobile element here is the clamp, one of these jaws of the RNA polymers that has to open for the double-stranded DNA to bind to the bottom of the channel, and that's where it opens, and then the clamp basically closes to lock it in place. So that's a clamp opening model. There's another model that suggests that the DNA opens outside the main channel, and then single-stranded DNA is much smaller than its double-stranded counterpart, and then it can bind to the channel without the clamp needing to open. Now, there are quite a number of issues here. A lot of this information that led to the models depends on ensemble averaging that cannot clearly identify complex kinetics that have many steps. And the structural snapshots that gave rise to some of these models, especially the clamp opening model, may actually not be on pathway, because this is not how the structures are observed. Plus, there may be key structures missing. And of course, when we discover a structure through structural biology, then we try to see whether it fits in this pathway. So therefore, here you can see a structural state that emerged in 2019 that shows that the DNA is in this particular state is open only halfway. So you have this partially open intermediate that may or may not be on pathway. So we wanted to address these conformational changes and to be able to see them at the single molecule level. In order to be able to achieve this, we need to be able to see the conformational changes of the clamp, to see it opening and closing where this happens, to be able to see the melting of the DNA, the unwinding and melting of the DNA in real time, and then to try to see these processes as they happen on the way to the final state. So here you can see some of the experiments that were done by a former graduate student in the lab, Diego Ducci, by looking at the conformation of the clamp, the mobile element that we're interested in. So in order to look at the conformation, again, we place two fluorophores at the tips of these jaws of the RNA polymers, and the open state of the clamp was expected to correspond to around 80 angstroms, and the closed state to around 70 angstroms. So this 1 nanometer change actually is quite significant for our method, and is readily observable if it happens. So therefore, we take these proteins, we place them on the surface, and then we just look at their dynamics and see whether we can capture the states. And to be able to do this, we immobilize the complex of the RNA polymers with the DNA on the surface using a series of interactions that anchor it on a surface that we can illuminate with light that gives us an evanescent wave that illuminates only the molecules that are bound to the surface or very close to the surface. So therefore, when we image now a certain area with single molecules, we get images like this, where you get on a camera, where you get bright spots that correspond to the signal of the donor in the green channel and the signal of the acceptor in the red channel. We have ways of aligning the channels in order to see which signal corresponds to, in the green corresponds to which signal in the red. And when we do this and we plot the intensities, we can actually see the fluctuations in thread efficiency that tell us that we are looking at conformational changes. So you can see here the green signal going down when the red signal going up. So now we have a high thread state, state where the clamp is closed, and states where this thread efficiency is low, presumably states where the clamp is open. Now, it's difficult to assign how many states you have by just looking at these traces. So we have systematic methods to identify the number of states. And actually, by looking at many of these traces and analyze them, we identified that there are not 2, but 3 conformational states. One of them being a partially closed state of the clamp. And of course, when we have all these duels on the various states, we can get the kinetics for the partial opening, the partial closing, the full closing, and vice versa. So we can recapture all these states. So this has been published, so I'm not going to go through this in great detail. But we also showed, apart from the fact that the clamp is dynamic, that you can use antibiotics to either lock the clamp in the open state, lock it in the closed state, or there are small molecules that are important for regulation in bacteria that actually lock it in the partially closed state. So that was quite fascinating, and also this proposes a mode of action for this particular antibiotics. And then we saw that after the DNA opens, the clamp remains in the closed state. Okay, so we established now a way of being able to see the motions of the clamp. So what about the opening of the DNA? So how can we do that? Well, for that one, we took advantage of the fact that one of our favorite fluorophores, Psi3, which you can see here, has a different, a very different intensity, very different quantum yield as you move from a double-stranded DNA complex to single-stranded DNA. So we see this huge increase by 120% as you go to single-stranded DNA. So therefore, when we looked at this transition from the double-stranded DNA to the open complex, we can see this a very large increase indeed that then allowed us to go and set up an assay to look at this in real time. So what do we do in this case? So we have the machine on the surface waiting for the DNA to bind. Once the DNA binds, you start seeing a signal appearing. And when the DNA melts and unwinds, your signal becomes brighter. And you can actually see this happening in real time at the single molecule level with millisecond temporal resolution. Here it is. That's the binding event. You're waiting. At this point, you can see the melting in real time. Now, this is fascinating because you can actually get the kinetics for melting, and then you know the time scale during which you can start looking at conformational changes from the perspective of the protein in order to time the various events. Okay, and you can do this by looking at molecules that are performing these motions along the pathway for the opening of the DNA. So here's the results that we got for a position that's very close to the downstream end of the bubble, the part that opens last, expected to open last. And when we looked at the time scale for opening for many molecules, we saw that this is described by this peak distribution that you see here, that actually implies that we are seeing, in this case, 2 rate limiting steps that are sequential. And the overall process takes about 7.5 seconds, and one of these processes are fairly fast and the other is slow. Then we ask the question, what happens if we actually place a fluorophore at the upstream part of the bubble? And then we did similar experiments, and we saw a similar distribution, but now the timing is 2 seconds shorter. But again, this is a sequential process. So that raised the possibility that you may actually have multiple rate limiting steps when you open the DNA. And we decided to look at this further. And we did an experiment where we increased the GC reach part of the DNA. So this is the one that actually that melts more difficult with more difficulty, because you have 3 hydrogen bonds that need to be broken per GC base pair versus an AT base pair. And when we did this and performed the same experiment, we saw that the melting time now increases to almost 19 seconds. So we made the breaking of the bonds in the double-strand DNA harder, and therefore the machine took longer to open the DNA. And then, in order to address the question whether there is a 2-step process here, we look at what happens for this GC reach promoter if you place the fluorophore at a position -9. So when if the whole thing opens in 1 step, then you expect to get around 19 seconds. If you have multiple steps, it will be much shorter, and that's what we see. We see that the melting basically takes only 6 seconds. So therefore, you have 2 steps, one that corresponds to the upstream part of the DNA and one to the downstream part. And then depending on how GC rich the sequence is in the downstream part, you have different time scales for opening. Then we wanted to answer the question, what happens to the clamp during this process? And then we inverted the way we did the experiment. Now we have the DNA on the surface, and when the protein binds, because we have these 2 fluorophore threads, we can get information about the status of the clamp, and then we can see whether this changes with time or not, and whether there is transient opening or closing of the clamp, as proposed by one of the models. So here we can see one of these events where the RNA polymerase binds, and we can see that the clamp is actually in the closed state and remains in the closed state. So in no point we can see transient opening and closing of the clamp. And when we look at the distribution for the first 5 frames, we see that the state is the closed state of the clamp. But when we look at all the frames, we can see that in a large number of the frames, we see 2 states, and the second state corresponds to a clamp that's even further closed, a state that we call the locked clamp. It's a new state that we discovered. So therefore, we don't see transient opening, and the clamp remains closed up to the point that it closes further. And here is an example where we can see a binding event waiting to the point that the clamp actually locks. And when we look at the time scale for this, you can see that's about 8 seconds. So it matches basically the time scale for the opening of the DNA. So therefore, our interpretation is that you have this 2-step opening of the DNA, the unwinding of the DNA, and when this takes place, then the clamp actually locks. So we put together an updated version of the model for the opening of the DNA. So we have initially a binding event, a partial melting that melts the upstream part of the DNA, then full melting takes place. The single-strand DNA is loaded, and the clamp closes to lock the DNA in place. So therefore, that is the case that for a bacterial promoter that has a consensus sequence, would be very interesting to see how this changes for different genes, and how this is regulated by proteins and small molecules, and of course, be very interesting to see what happens in different organisms. So far, I presented to you an example of an in vitro single molecule measurement where we have a lot of control. We can modify the surface the way we want. We can link our molecules with sophisticated ways. Or if you do an optical tweezers experiment, for example, you can trap beads with DNA between them, and RNA polymers can pull the beads, and you can measure forces and see single steps. So fascinating work, but you're missing the context that comes with the biological medium of the live organism. And of course, when you have this, you have a lot of complexity. You have other biomolecules that may modify what you're studying. There are other processes where your molecule can contribute. There is the macromolecular crowding that takes place because of the high concentration of proteins and other macromolecules in cells. And of course, you have compartments, even in bacteria, that may change the interactions that are taking place. And stochasticity, because many of these molecules, like the DNA, exist in very low copy numbers. So there are distributions of properties that can lead to many different phenotypes in single cells. So we wanted to be able to look at single molecules in single cells, and that was a project that was spearheaded by a fantastic former student in the group, Matthew Stracey, who looked at how RNA polymerase moves inside bacterial cells. So when the RNA polymerase is searching for a gene, it's taking long steps. It's diffusing actually fairly rapidly, but when it binds to the DNA and copies its information, it is essentially immobile, or moves extremely slowly. So therefore, by just looking at the mobility of the RNA polymerase, we can see basically where it is, where the genes that are transcribed are in the cell. So, and to be able to do this, we used a property that has been exploited in single molecule super resolution methods, where you can photoactivate a molecule. You can observe it for a short time, and then you bleach it, you lose it. And that's important because if you want to look at all the RNA polymers in a bacterial cell at the same time when they're in an on fluorescent state, then you just see a blurry image. You don't see a single molecule because there are 3,000 of them. And here you can see the image of a single molecule in a single bacterial cell. But if you just turn only one of these molecules on at any given time, you see an image like this, and then the molecule may move or may actually appear stationary. So therefore, every time you do this, here's an example of a movie, you can see a molecule. Here you can see that the molecule moves fairly significantly, and then it bleaches. And every time you get a track, and then you can put all these tracks in single cells. So you can see hundreds or thousands of molecules in single living bacterial cells. And that can give you lots of information. It can give you some idea of the copy number, how many proteins of a certain kind you have in a cell, and you can look at the distribution in single cells. You can look at where the molecules are in the cell and how they're moving about, and where they are relative to other proteins or other structures in the cell. And also you can, by looking at the mean square displacement, an index of mobility of molecules versus time, you can see how mobile they are, and also get some information about their containment that will be witnessed by this saturation in this particular plot. And the other thing that is fascinating is that you can actually monitor as molecules are moving, and then see when they're binding to a certain target, how long they stay, and therefore you get information about timing and the presence of the targets in cells. So in the case of RNA polymers, we performed these measurements. We look at, you see here, many trajectories, 70,000 trajectories, and we got this wide distribution that can be fit by two distributions. And we do this because we understand this red distribution very well from control experiments. So therefore, we can see that about half of the molecules are bound and half are diffusing. So these are the molecules that are transcribing or binding to the DNA. That's our interpretation. And half of this of the total molecules are searching for the genes. And what is fascinating is that if you use a certain threshold to color your tracks, either red for not moving fast or blue, you can see that distribution is very different. You can see that the molecules of diffusion seem to be centrally located and occupying what you expect the region you expect that will be occupied by the chromosome, and the red molecules seem to crown essentially this structure, and these are the ones that you expect that will be copying the DNA into RNA. If we now put an antibiotic in the cells, that basically stops this process and just keeps the molecules that are on the promoter, you can see that the distribution changes massively, and as you expect. And this actually gives you an opportunity to see how antibiotics function, and also to see whether some cells are resistant to antibiotic, because this is expected to give you a very different profile. So how does this distribution that we see for these proteins that interact with DNA actually compares to the location of the DNA in cells? So here, this information from a single cell. We can stain the DNA in live bacterial cells, and we can look at the location of the mobile RNA polymerases. And you can see here that the mobile polymers essentially stain the DNA perfectly. It's almost like a DNA stain. So this is not a fit. This is actually a superimposed image, superimposed curve of the DNA on the distribution of the RNA polymers. And something similar here also if you project on the x-axis and the short axis. However, when you look at the bound polymerases that are expected to transcribe genes, you can see that the profile is very different. Now you can see that the polymerases tend to be in the periphery of the DNA and the nucleoid. And this is something we've seen when we looked at hundreds of cells. Basically, this recapitulated the fundamental picture that we get from a single cell. We also wanted to see whether we can perturb this distribution by changing the amount of DNA in a cell and the region that it covers. So you can use mutant E. coli cells that replicate DNA less, and you can condense their DNA, and therefore you can concentrate the DNA in this part of the cell and leave these ends, the end caps as we call them, free of DNA. And now what you can do, first you can see how this compares to the cells that have not been perturbed, and you can see that the polymerase becomes a lot more mobile. So all this bound population decreases very significantly. And the other thing that you can do, and I'm always fascinated by this result, you can actually look at diffusion only at this part of the cell that is free of DNA. And when you do this, you can see the diffusion is much faster. So, and if you do corrections that account from the fact that you have containment of these molecules in a cell, and there is some error for localization and precision, you can see that in the absence of the DNA, diffusion of RNA polymers is fairly fast, but in the presence is much slower, which suggests that actually 85% of the time when the polymerase is searching is spent non-specifically bound to the DNA. So we pursued this further recently and looked at the behavior of many DNA binding proteins that have different functions, but all work one way or another on the DNA. Because the DNA for these proteins is doing many things. It's the reaction substrate. So for example, if there is DNA damage, there is a protein that will recognize the damage and fix it. But it's also the search path, because it's been shown that the DNA binding proteins are searching, binding non-specific natural and perform searches in order to find their target. And then, of course, it's a mesh. It's like a sort of basket that needs to be explored and can act as an obstacle to diffusion. So here is an example of various proteins that interact with different types of substrates. You can see here we covered RNA polymerase. DNA polymerase 1, for example, will bind to a gapped DNA substrate, and then different binding proteins will bind to different types of DNA. But in all cases that we examined 11 such proteins in a paper that was just published in Molecular Cell. You see two populations. One population that appears to be bound stably to the chromosome, and one that contains both the non-specific DNA binding and a certain fraction of diffusion of the DNA. So like three-dimensional diffusion of the DNA and then non-specific binding the DNA. So that's what we see again and again for all the DNA binding proteins. So we wanted to see whether we can find a way to look at DNA-free diffusion without having to play the tricks of basically coming up with bacteria that have minimal DNA. And to do this, we introduced two sites on the DNA where a protein can bind, make cuts, and then another protein will digest the DNA. So this way, you can actually digest the DNA in about 90% of the cells within 90 minutes. So therefore, you have lots and lots of bacteria that have no DNA. Now you can monitor the diffusion in this bacteria and then get how RNA polymerase diffuses in the absence of any DNA and the absence of the chromosome. So now all this distribution that contains these two species, the bound and the diffusion, become distributions that have only one species. And now this species is actually very fast because it represents only the three-dimensional diffusion of these proteins in bacterial cell. And having these numbers, the diffusion coefficients and the relative abundance, allows us to calculate the fraction of the time that these proteins are bound, are not specifically bound to the DNA. And as you see here, these three examples is always over 50%. So therefore, the non-specific binding of the DNA dominates the search process for these proteins to find their targets. And here you can see how when we plot the diffusion behavior, diffusion coefficient of these 11 proteins versus their molecular weight, we can see that there is no trend, not the trend that we're expecting if we just look at the behavior as described by the Stokes-Einstein law that predicts the diffusion coefficient as a function of the molecular weight and the viscosity of the medium and the radius and the density of the protein. So in the case, however, that we look at the DNA-free cells, we can see a linear relation now that the DNA has been removed. So there's a good match to the Stokes-Einstein law, but the exponent here, instead of being basically 1/3, basically a cubic root of this factor here, is 0.75, which means that even when you digest the DNA, there's other stuff in the cytoplasm inside cells that slow down diffusion and cause effects that resemble sieving. So just to conclude this part of the talk, so I showed you how we can use this tracking of single molecules to get the spatial distribution of mobility. I showed the importance of this non-specific interaction, both for RNA polymers and for other DNA binding proteins. And I also showed you that the polymerases that are performing transcription are actually moving at the periphery of the nucleoid. And I didn't cover the presence of clustered RNA polymers due to the lack of time. Now, only have a few minutes, so I'll go through quickly some work that we've done on viruses using single molecule imaging methods. That's work of two brilliant people that worked in my lab. Nicole Robb, who was a former fellow in the group, and now she's an associate professor at the Warwick University, and Nicolas Cialis, a brilliant undergraduate, now graduate student in the group. And the idea is to use the power of single molecule imaging combined with deep learning to be able to detect viruses and identify them rapidly. So the whole project started with the ambition to be able to detect viruses through an interaction with molecules that bind specifically to the surface of enveloped viruses. And here you can see a cartoon of the flu virus. So the idea was the following. If you have a molecule like an antibody or an aptamer that binds to one of the surface proteins of the flu virus and look at the diffusion of this labeled particle, you expect this to diffuse slowly because the particle is fairly large. The diameter is 120 nanometers. However, if you don't have particles, then the antibodies will move extremely quickly, and therefore you will see a very, very different signal. So in the process of actually doing this work, we discovered a new interaction between single-stranded DNA and the envelope of these viruses that's mediated by calcium ions. And this is non-specific, so therefore it can work with any envelope virus. And in order to be able to visualize the particles, we used approaches that are similar to the ones that we use to study diffusion in bacteria. The particles are labeled so we can track them, and then with tracking analysis, we get information about their diffusion and therefore about their size. So, and this is how the movies look. So you add basically calcium chloride, high concentrations, and fluorescent DNA, and with 1 minute, you get basically the viruses appearing and diffusing. And then you have lots and lots of events that you can analyze. First, we showed that the signal is very specific. So if you don't have virus or calcium chloride DNA, or you remove the calcium ions, you get nothing. But if you have these interacting components, you get lots of particles. You can track them. You can see here some of the tracked viruses, and then you can recover the dimensions of the virus particle, and you can count the viral particles. And this is very fast. As I said, you add the virus, a sample that contains the virus in a solution of calcium chloride DNA. You mix up and down. You put on a well on top of the microscope and record movies. And even within 1 minute, you get lots and lots of events. And then you can see here how the number of particles that you detect changes with time. Within 1 minute, you have more than 50% of the signal. So it's very, very rapid. Then we tried to analyze the mechanism of the interaction. We saw that the binding is specific to calcium. It worked best with DNA molecules, but RNA also showed a signal. And you require a minimum length of 20 nucleotides or longer. And very importantly, the charge for the interaction needs to be negative. So when we did experiments with neutral or cationic vesicles to simulate viral particles, basically we got no signal whatsoever. So therefore, our interpretation is that you have this, in this case, you have this negatively charged virus envelope, and the negatively charged phosphates of the DNA and calcium basically acts as a bridge to put many of these fluorophores on the surface to make them very bright so you can detect them very quickly. You can do this also on the surface by placing molecules on the surface. You can use antibodies that label on one color and the DNA is on another color. You can capture these particles on the surface, and then you can do two-color microscopy to be able to see the particles. So we concluded the work and we published it in November 2019 in Scientific Reports. And then, as we all know, our life changed in 2020 because another envelope virus came into prominence and made us all very miserable. It's SARS-CoV-2. So we wanted to see then whether we can use our method for labeling and another method that was developed to do virus identification to be able to detect and classify CoV-2 particles. Now, that was important because if you look at the topography and sizes of common coronaviruses that cause to humans just a common cold and influenza, you can see that the topography and the sizes are fairly similar. So therefore, we needed something to be able to tell subtle differences between the properties of these particles, and that's where Nicholas came, because Nicholas was working on finding ways to use deep learning to tell the difference between different strains of influenza by using imaging and machine learning. So this is the idea that was promoted by Nicholas. So let's say they have two viruses with different size and with different surface properties. When you label them with this calcium labeled method and you place them on the surface, you can image and get individual signals from individual particles. Now you can feed them in a deep learning network where you of course have labeled the particles from virus X in a certain way, and then you also annotated the other virus with that corresponds to the different virus in a different way, and then you feed this in a convolutional neural network that then basically tunes its parameters in order to recognize specifically the particles from a different virus. And when you do this and you have trained your network, you can have unknown samples that can be examined by the trained network, and then every particle will be classified either as virus X or virus Y with a certain probability. And of course, because you have many particles in samples, then you can also identify the particles, the sample that contains these multiple particles. So the experiments are fairly similar to the ones I described before. Again, mobilized particles on the surface and two-color imaging for two DNAs that are binding to the viral particles. So we do this with an animal coronavirus, chicken coronavirus, and we do this also with also controls. And then we have ways of identifying the particles that are colocalized and therefore are likely to correspond to viral particles. And then we will have some similar particles in a negative control that we need to differentiate from. It's a very fast process. Here you can see the microscope scanning from field of view to field of view. So this is in real time. So you can basically get tens of thousands of particles in just it's a couple of minutes in automated fashion. So when we did these measurements now with different viral particles with a negative control, we could see that if you look at the various properties of the image, like the maximum intensity, the area of the particle, and also the asymmetry of the particle, then we can see subtle but clear differences between viruses and the negative control. And that's what then is used by the network to be able to classify the particles as a positive or negative, or as belonging to one virus versus another. In order to evaluate the performance of the network, you have to prepare this confusion matrix, as it's called, where this is the actual property of a certain particle, and this is what is output by the network. And clearly, the best performance is to get very high true positives and true negatives, because this will correspond to high sensitivity and high specificity. So this analysis is done for different particles, and the probability reflects just one of these particles. But when you have a clinical sample, for example, you have many particles. So therefore, if you use these images, then you can call the clinical sample with high accuracy. So, but when you look at the particle level, here you can see that in the case of the animal coronavirus, we can tell the difference from a negative control with very high validation accuracy, over 90%. This is a lab-grown strain, however. When you go to clinical samples, that clearly they're more complex, and we compare the coronavirus, the common human coronaviruses with a negative control. We get a validation accuracy that approaches 80%. That's again per particle, not per sample. And even in the case of SARS-CoV-2 clinical samples, we get a 70% validation accuracy. But again, this is per particle. So I'm emphasizing this per particle because if you look, let's say, at a sample that has been shown to be positive by RT-PCR, and you perform our assay, you will have many particles. So these particles in this positive sample has been classified as 148 being positive and 25 as being negative. So therefore, if you look at the probability of this particular sample being positive, you can see the probability is extremely high, and vice versa, the probability of a sample that has only 25 out of 173 being negative is very, very low in terms of being negative. So therefore, this way we can identify and call the samples being positive or negative. So I'm almost done. Just to conclude this part of the talk, I showed you how cation-mediated labeling can detect viruses very, very quickly, and our deep learning approach that can achieve over 90% accuracy per particle. Still working on improving this accuracy. The detection, of course, is very fast. What we're trying to do now is to determine the sensitivity and specificity for a large number of clinical samples. We're trying to see whether we can capture different variants with this method, and trying to develop an affordable detector to be able to do this analysis. This is something that's pursued very heavily by Nicholas and Nicole. Of course, this is something that's not limited to coronavirus and can be used to detect other viruses. So I'll end here. I will thank my wonderful group that made all this work possible, especially Abhishek, who contributed in the in vitro analysis of the transcription initiation mechanism, Nicole and Nicholas, who spearheaded the efforts on viral detection, and Matt Stracy and Stepan Uphoff, who did the wonderful work on single molecules of DNA binding proteins in bacteria. And if you're interested in joining our group, we have a postdoc opening on deep learning approaches to look at antimicrobial resistance detection. So I'll stop here, and I'll thank you for your attention.
Thank you so much for this very exciting and intriguing talk. Now we go over into the question-answer session. Please ask questions. Send in a message through chat button, or just raise your hand. Achilles, I have a question. You have
Sure,
very nicely explained indeed how to distinguish between different viruses. The method, of course, is universal by nature. Fluorescence spectroscopy, single molecule imaging. But you at the end mentioned also about variants. So the same virus, not different virus, but the same virus with mutations. How does this work for this case? Can you now, for example, detect using these methods some variation? You know, of course, African variants or England variants or Brazilian variants of COVID coronavirus. Can you distinguish between them using this method?
So, you know, you wouldn't necessarily expect that this is going to work necessarily, but the way that the work had started was by looking at different strains of flu that, again, you don't expect them to be hugely different, but there was very good discrimination between different strains of flu. So when variants started appearing, then that was something that was tested in the lab, and some discrimination has been shown. I mean, again, this is work in progress. Now, the variants may have different charge distribution and different behavior in terms of the interactions between particles, and this is something that seems to be being picked up by deep learning. So there is some discrimination, and then you can change the conditions to try to increase the difference, the initial difference that you see. Yeah. Whether this is going to be usable as an actual way of being able to tell the difference at a large scale for clinical samples, I mean, that remains to be seen.
No question, please. have one question in chat. Achilles, can you read them?
Let me see. Yeah, okay, there you go. All right. Question about bacteria. Can known bacteria develop resistance so that known antibiotics for them become ineffective? Yes, they very much can do this, and this is one of the major problems that we will be facing. We're facing now, and we'll be facing even more the years to come if we don't take action. Right now, there are 700,000 deaths a year in the world because of the microbial resistance. So for you go to the doctor, they prescribe antibiotics and they're ineffective. And if this happens in diseases where the effects of antibiotics need to take place very, very quickly, like in the case of septicemia and sepsis, then the results can be catastrophic. And because we are overusing antibiotics, especially broad range antibiotics, because it's simpler for the doctors to prescribe and it's faster, then essentially we are helping bacteria to develop resistance, and therefore we need to do something in terms of being able to detect their resistance and then develop new antibiotics, because otherwise the numbers will go from 700,000 that we have today to 10 million by 2050 if no action is taken. So another thing to worry about, but there are many groups that are working from different perspectives to make sure that this doesn't happen. I have to say that some of the techniques that we have been developing for coronavirus came from the need to develop better ways of detecting antibiotic resistance. But now that we have gotten better in some of these techniques because of the pandemic, now we'll go back and use these improved tools to look at how we can tackle resistance in bacteria.
More question, please.
It's probably getting late for some of the people. It's been the time is there, so eight something, right?
So if no more question to what do you think, Achilles? Can you stop? Can we stop here?
Yeah, absolutely. Well, thanks for attending the talk. Thanks for your questions. Okay.
This is indeed not question. Thanking you for excellent talk. So I agree with this question. So is there other scientific questions? So it seems to me that no questions. So if you don't mind, we can stop here.
Yeah, no worries. And
thank you very much once again. That was very, very excellent.
Thank you. And our website is available in case that people are curious to find out more.
Yeah.
So stay well and see you sometime in the future. Take care.
You too. See you.
Bye-bye.
Thank you. Bye.